While this was more thoroughly covered in lab, I do want to do a quick run through blood types, blood typing again. The idea of blood types is based on antigens, and this is a topic that is very important when discussing immunity overall. Antigens are chemicals, substances that can trigger an immune response. Most of them are going to be a protein of some kind. There are other types. Often they may be a glycoprotein, a protein-sugar combination. But you will find antigens, maybe they could be free-floating, but a lot of them are going to be on the surface of cells. And sometimes there are chemicals that could be considered antigens that are just put on cells. Cells have them, and they are ways to identify that the cell is of a certain type, belongs to an organism. Right? So these surface antigens. So having the correct surface antigens would be an indicator to the body's immune cells that, oh, I'm a part of the body, please don't kill me. If it is not recognized, however, then there can be an attack here. The body typically does not respond to self-antigens, as I mentioned, but if they do, then congratulations, you have an autoimmune disorder. That's actually not a good thing, of course. The immune response does involve antibodies, and we'll talk about how antibodies are made by particular lymphocytes later on in the immunity chapter. Antibodies have a lot of different jobs that they can do. One thing that they do is simply just grab on to the antigens and hold on to them, causing them to bind together. And as this causes a clumping, that's called agglutination, as they clump together. When discussing blood cells, and specifically red blood cells, there are three antigens to look at. Two of them are genetically related and form what's called the ABO group. The other one is not genetically related to the other two. It's called the Rh factor. All right, so with the ABO group, we can have cells with antigen A only, cells with antigen B only, cells with both, because this is a genetic thing called codominance, where if both the A and the B allele are present, they will both be expressed. Or if neither the A nor the B allele are present, we call that type O, kind of like 0, but we'll call it O, the letter, because neither antigen A nor antigen B will exist. All right. Now, genetically, you can't be alive without actually getting some exposure to antigens A and B, and what that really means is that if you do not have a particular antigen as a self-antigen, your body will make antibodies against it. So if you're blood type A, you've got anti-B antibodies. If you're blood type B, you've got anti-A antibodies. People that are AB don't have either. People with blood type O will have both, because neither will be a self-antigen. These antibodies are of a type that cannot cross the placenta. So this will not have any bearing necessarily on an interaction between a mother and the developing fetus in utero. So the cross-reaction of red blood cells and opposing antibodies, as I mentioned, the clumping, is agglutination. This can cause some problems as small blood vessels can get clogged, but also these blood cells will eventually burst. That's called hemolysis. And having this free-floating hemoglobin in circulation can be dangerous. It really damages the kidneys. When the body does break down red blood cells, it has to do so in a very specific way, not only to recycle the iron, but to prevent damage to the kidneys. It will only be done in the spleen and the liver, having it happen just in circulation. Very good. All right. So that's the ABO group and a little bit about just what will happen if there is a cross-reaction. But let me talk about the Rh factor. So the Rh factor is a third antigen, and it was originally discovered in rhesus monkeys, hence the Rh in there. Most people have it, some people don't. All right, something like 3/4 of people are Rh positive, and about a quarter not. Rh negative individuals don't necessarily have anti-Rh antibodies, however. You won't just get exposed to it in the environment like you would A and B. An Rh negative mother, however, can become sensitized to the Rh factor if she gives birth to an Rh positive offspring, right? The sensitization means the system will now recognize it. Initially, the blood supply won't mix really, all right? So the placenta will kind of keep things separated as it's delivering nutrients, but there's crossover at delivery because of the bleeding that will occur there. Now the Rh factor will actually enter the mother's system, and she will develop these antibodies. What does it mean for the kid? For this kid, nothing. All right, because it's having a delivery, so how it goes. Problem is if there's a second kid who is also Rh positive, because the antibodies that are made against the Rh factor actually do have the ability to cross the placenta and begin destroying the cells, the red blood cells within the fetus. And so this is called hemolytic disease of the newborn, or erythroblastosis fetalis. This is definitely not a good thing. All right, so what do we do against this? There is a defense. There was a serum that was developed, and it can be given to a mother around the time of birth. It is called RhoGAM, and I will write that at something like this, I believe, in terms of how it's written. And this particular serum is basically going to bind up the Rh factor so that the mother never gets sensitized to the Rh factor. And will never develop the antibodies, so subsequent offspring are not going to be placed in danger. And the very last thing about how you would actually determine the blood type is all about an agglutination test using various sera. So with the anti-A antibodies, the anti-B antibodies, and the anti-Rh antibodies, and looking to see if there is agglutination that occurs. If agglutination occurs, then you know that particular antigen is present. So here, all three antigens are present, AB positive. Here, none of them are present, O negative, and so on.